Hey guys, I'm Jolie, and today I'll be talking to you about the field of nanomedicine and then briefly about a pretty cool process known as electroporation. So, a little brief intro about myself. I'm actually a bio major currently on the pre-med track, and to find some inspiration for this mini project, I was looking into different interconnected topics within the fields of biology and physics, and I found a lot of cool stuff. But to be frank, I'd say that a lot of it was out of my depth. So I just kept looking, and eventually I decided to go with nanomedicine because that's something that I find both interesting and highly relevant to my future career path. So without further ado, you're probably wondering, what exactly is nanomedicine? Well, as the name suggests, to put it simply, it's really just the use of nanotechnology in monitoring and controlling the human biological system. It's widely used to diagnose and treat diseases at the atomic and molecular level. It's like thinking of the human body as sort of a machine that can be fixed with the use of these extremely small nanoparticles. And really, there are all kinds of nanoparticles that are used as sort of drug delivery systems within nanomedicine. You find polymer-based nanocarriers, inorganic nanoparticles, viral nanoparticles, drug conjugates, and lipid-based nanocarriers. And this last one's actually something that I'm more familiar with. So when I was doing research over at the University of Tor Vergata in Rome last summer, my department mostly worked with liposomes, which makes sense because I was working in immunology, and it was a kind of bench research involving the manipulation of liposomes to eventually be used as a form of drug delivery when dealing with the multi-drug resistant non-tuberculosis Mycobacterium abscessus, which commonly causes chronic lung infections. This, of course, is exacerbated in patients who already have underlying lung disease, such as cystic fibrosis, which is more precisely the demographic that we were looking at. But I'm going to stop right there because the rest of this video is actually going to be more about electroporation, not my research, which is really just one of the many processes in which nanoparticles can be used for research, both clinical and non-clinical. I guess just think of it as kind of a subset within the much larger field of nanotechnology and medicine. So there's the technical definition from Oxford Dictionary, but to break it down, it's really just the use of electricity to introduce foreign DNA into a host cell by creating new pores within that host cell membrane through those electric shocks. And of course, the entire field, as I've been saying, involves many types of physics as a whole, but electroporation is where I found a connection more specifically to Gen Phys II. This technique, also known as electropermeabilization, is so cool because, again, it involves the use of an electric field, which we learned all about this semester. Basically, by administering short pulses of high voltage current, an external electric field that surpasses the electrical capacitance of a cell membrane is applied to the host cell to increase the electrical conductivity and the permeability of that cell to different kinds of drugs. And electroporation can be used for not just drug, but also gene and vaccine delivery. And as I already mentioned, these pulses are short. They're like millisecond long electrical pulses that can create those temporary pores to drastically increase the cellular uptake of those target particles. But it's actually kind of different for drugs and genes. If you're working with drugs, you can administer the drugs before or after the pulses have been administered, but with DNA, it's a lot different. Instead of short high voltage pulses, you're actually going to want to use long, low voltage electrical currents, and the DNA would also have to have been previously administered prior to the pulses. So examples of that would be the synthetic DNA vaccines that are administered into the skin or muscle cells. And you're probably wondering, like, why can't DNA just go through? I mean, drug delivery seems so much faster. If you think about it, it's really just simple diffusion occurring once the membrane has holes created in it. Well, that's because DNA is actually way too big a molecule to enter through those hydrophilic pores through just simple diffusion alone. It's also very negatively charged. So again, if you think about it, this isn't why like DNA is more complicated. It's just something cool. Because it's negatively charged, as we learned in physics, it's going to be attracted towards the positive end of the electric field. Yeah. And it's also, this whole thing is actually very similar in theory to gel electrophoresis, if you're familiar with that process. Lastly, one important, so yeah, again, this is right here. This right here is just showing you everything that I just explained. That's showing why DNA can't really go through. Yeah, lastly, one important takeaway from all of this is that electroporation can actually be used in transfection and transformation. Transfection is the transfer of DNA into mammalian cells, whereas transformation is the transfer of DNA into plant, yeast, or bacterial cells. This right here just shows bacterial cells. And yeah, and a large part of what I talked about today involves transfection, but as of yet, synthetic DNA vaccines have only been approved for the use in animals and not humans. That being said, we're learning more and more each day and implementing new changes into our lives. So I think this is a really cool and innovative application of physics within the field of medicine. That's about it. Thanks for listening, guys. Have a good day. These are my sources. Thank you.